All right. Welcome, everyone. So today is the last lecture of our disease module and population genetics module. So basically, we talked about population genetics and sort of the basic variation in DNA sequence. We talked about disease associations and how that genetic variation correlates with phenotypic variation. Then we talked about EQTLs and how intermediate molecular phenotypes allow us to explain these, the mechanisms underlying these broad phenotypes at the organismal level. Today, we're going to talk about a topic of great recent interest, namely that of heritability. So we're going to talk, first of all, about what is heritability, how do we define it, what are the key concepts. And then we're going to talk about how heritability allows us to understand genetic architecture of complex traits, and then how we can go, from genetic architecture to systems biology, basically understanding what are the pathways underlying these broadly heritable traits, and then talk about linear mixed models and how we can use them to actually start predicting phenotypes. So genome-wide association studies have had tremendous success. They've discovered thousands of genetic loci underlying complex traits. But there's some broad lessons that we can learn by sort of stepping back and asking, what have we learned from these studies? The first thing we've learned is that we haven't actually found all causal loci. Namely, the known loci explain very little phenotypic variability. And most loci affect transcriptional regulation. They don't tag coding variation, but they tag non-coding variants. And for that first point in particular, we'd like to have a quantification for that. We'd like to know, well, how much is there to explain and how much could we possibly explain? What is the upper bound and when will we be done? And that's a hugely important problem because the question is, do we go after the biology of the known loci? Do we just go and discover all the loci first? Will we ever be done? When will we be done? And so forth. And we need some tools for doing that. And the main tool that the community has been using for this is understanding heritability. So what is heritability? Heritability is basically a way of, it's basically the fraction of phenotypic variance that you can explain from genetics, okay? And to get at that fraction, what you need is to actually decompose phenotypic variance into its sources. If you have a phenotype like height, and you look at how much does it vary in the population, you can basically say, well, there's a certain, variability in that phenotype. Like that phenotype is basically, varying by a certain amount in the population. We can study how much does that vary. And then that phenotype is driven by two components. On one hand, inherited genetic components. On the other hand, environmental components. And what we're going to do is basically try to study the fraction of that phenotypic variability in height that is explained by genetics and how much of that fraction is explained by environment, okay? And then, of course, this would also be including the covariance between genetics and environment. But for now, we're going to assume that there's no gene-environment. So what genetic heritability is about is taking this phenotypic variance and then splitting it up into its genetic component and its environmental component, okay? Who's 100% with me so far? Awesome, great. So there's a problem here, which is if I introduce a milk, delivery program for newborns or something, and that causes a change in the height of the babies that were administered this extra milk, for example, in a population that is otherwise nutrition limited. Then what you're going to see is that there's in fact increased variability in height. And that increased variability will basically come from my milk program, from the environment. Suddenly, the genetic heritability of height will go down, right? Because I'm increasing this blue component. Conversely, suppose that I now realize that height can be more accurately measured. Instead of measuring height, at adulthood, we should measure height at 14 years of age. And suddenly I realized that, in fact, that measurement gives me a tighter definition of age, of height. And that, in fact, reduces the overall variability. So then the purple line will go down. And if I can explain a big chunk of it with a red, then suddenly the heritability of height will go up due to having more accurate measurements. Who's with me on those two last points? Therefore, heritability is not something fundamental about a trait. It is a relationship between my ability to actually measure that trait, my ability to control how much of that is environmental, and the actual effect of the environment on that trait. Everybody with me so far? Cool. So this is basically what today's lecture will be about. Number one, how do we quantify, heritability? And number two, how do we use that quantification to now start partitioning heritability, basically explaining how much of that heritability can be explained using different fractions of the genome and using that to gain insight into the biological processes contributing to that. So here's one example. There's, one causal variance that determines some phenotypic output. Now there's three possible genetic values in the population. Now the intuition is that V of G is the variance of the mean phenotype across different genetic values, and VE is the variance of the phenotype for the same genetic value. Okay, so one way to get at how much of that variance is genetic versus environmental, you can basically say, well, genetics is clearly that component here. It basically tells me, what's the difference between the means, and then the quote unquote noise within a given genetic value. This is the non-genetic component, and that's basically effectively the rest, which we call very broadly as the environment, which could be all kinds of factors. Okay? A simple example. Awesome. Any questions? Great. So now we're going to start understanding these the components of the genetic variance. So basically, if you have, some kind of genetic score and some kind of phenotypic output, basically V of G, this variance according to genetics, can be further decomposed into multiple components. So the genetic component can be split into an additive component, a dominant component, and an interaction component. The additive component is basically what we're going to be focusing on throughout, this course and also throughout most of genetics, because it basically corresponds to some kind of linear model. I can basically model the additive effects of all of these different genetic variants using a very simple additive framework. And if we add more causal variants, the phenotypes become closer to Gaussian. In fact, with as few as 5 or 6 genetic loci, when you look at the inheritance patterns and you look at the distribution in the population, it starts looking quite Gaussian. As you start increasing that number, that distribution approximates more and more a Gaussian distribution. And that was the key insight of that Fisher 1918 paper that we talked about a couple of lecture groups ago. Namely, the fact that things that appear to be, distributed as a continuum in a population can in fact be perfectly modeled as a sum of multiple Mendelian traits whose phenotypic output basically becomes a Gaussian as you add more and more of those traits. And then you can further decompose these interactions, and you could include variance due to de novo mutations and so on. So basically, the additive component is the linear model. The dominance component are the places where one allele dominates versus another allele, so it's not purely additive. And then the interactions are places where basically multiple alleles in multiple loci in fact interact with each other. So what do we have so far? We basically have the phenotypic heritability that can be split into genetic and environmental, and then the genetic heritability and variability that can be decomposed into an additive, a dominance, and an interaction component. So we're going to be defining two terms, so big H squared and little h squared. So big H squared is the broad sense heritability. So that basically says what is the heritability captured by all genetic factors, which includes, additive, dominance, and interaction. The narrow sense heritability basically says what is the fraction of the phenotypic variance, not of the genetic variance, but the phenotypic variance, that is captured only by the additive components. And that's basically what we can, simply add up in linear models. So if this is all of the variance, then this is the genetic variance. This is the additive component of that. And then there's the dominance effects and the interactive effects. And then big H square is basically this over that. And then little h squared is this over that. Okay? So narrow sense heritability only captures the additive effects. And again, there's a big ongoing debate about the relative importance of additive versus other effects, the role of selection and so on. Yeah. Sorry, your question is how do we get VA? Yeah, so I mean, that's why we're sort of narrowing down our definition, because we're going to be looking specifically at these GWAS hits, basically asking how much do they add. And the easiest way to do that is to basically lump them all into an additive model rather than think about dominance or interactions, which can sort of, on the modeling side, we are simply unfit for modeling with current data sets and current understanding. And we'll simply kill our statistical power if we try to sort of learn these interactions and dominance effects de novo. So that's sort of what we're gearing towards, basically saying, listen, we can't model dominance and interaction for now, so let's just focus on the stuff that we can actually model. Say it again, Lilo. Exactly, exactly. So that's what we're going to be doing for this lecture and for most of the field today. Okay? So why do we even bother studying heritability? So basically, on one hand, it basically quantifies what is the importance of genetics versus environment. It teaches us about genetic interactions and genetic architecture. How many causal variants are there? What are their effect sizes? What are their allele frequencies? And it can actually tell us something about whether we should be looking for more genetic variants and how much more can they possibly explain. And also the narrow sense heritability is a fundamental parameter which is needed for phenotype prediction. And it is the theoretically best possible prediction performance with a linear model. Again, we're going to be focusing on linear models. Okay? So that's why we're studying heritability, because it basically is just fundamentally an upper bound on how much we could possibly learn from genetics. And it also teaches us about, models that may be inconsistent with this. Who's with me so far? Good. Any questions so far? Okay. So how do we estimate heritability? So basically, you can estimate heritability using relatives. You can basically say heritability relates phenotypic correlation to genotypic correlation. If two individuals have the same allele at every one of their causal variants, then you would expect them to have the same phenotype if everything was genetically driven. So this Haseman-Elston regression fits a linear regression of phenotypic correlations against genotypic correlations. You can basically say how correlated are individuals at different distances from each other. And that you can derive from family relationships, for example. Monozygotic twins, they would be at 100% similarity. Siblings would be at 50% similarity, first cousins, and so on and so forth. Okay? So you basically have this decreasing similarity at the genetic level. And then you can ask just how similar are these individuals at the phenotypic level. So one way to look at that here is that where we're going to basically looking at pairs of individuals, we're going to be asking, what is the similarity at the phenotypic level and what is the similarity at the genotype level. And then H square is going to be effectively the relationship between how similar are two individuals at the phenotype level and how similar are two individuals at the genetic level. So I can estimate H square as the ratio of basically phenotypic similarity versus genotypic similarity. Okay. So you can estimate heritability from GWAS as well. So basically this is, simply looking at how much individuals for whom I know the genetic relatedness level, how much do they vary phenotypically? And then you can sort of build a regression there. And then, the slope of that line tells you about heritability. Another way to do it is to basically start from the bottom up, from the stuff that we can actually measure. And basically say, well, I don't know about dominance and I don't know about interactions, so I'm just going to look at additive heritability. And then I'm going to ask, well, how much of that phenotypic variability can I explain from all of the genetic variants that I have and their effect sizes? So we're going to build a linear model that basically tells us this is the genetic component, which is going to be, you know, some function of my genotype times some kind of effect size, you know, for each SNP. So we can estimate the effect size beta of every SNP from genome-wide association studies. Okay, that's what GWAS gives you. So basically, GWAS basically tells you, you know, this variant has a 1 millimeter effect on my height or 0.5 millimeter effect on my height. Okay, so you can basically take that beta, that effect size of every genetic variant from GWAS, and then put it all together into a linear model that basically tells you how much genetic variability can I explain directly from the SNP that I have discovered. And this variance explained by every SNP depends on both its effect size and its minor allele frequency. Because if a variant has a huge effect size, but it is found only in 1 person every million, that doesn't explain too much of the heritability in the population. Okay? It basically explains a lot of heritability for the people who have it, a lot of the phenotypic variability of those people who have that variant, but very few people have that variant. So that's why the overall heritability depends on both components. Who's with me on this part? Great. So, you know, this variance basically is effectively, you know, some component of the minor allele frequency and then the effect size of every SNP. Okay? And if you do this with genome-wide significant SNPs, you usually have H square, a narrow sense heritability explained from GWAS, on H squared G from GWAS, that is dramatically smaller than the theoretical H square that I would get from, you know, basically from the previous equations that we saw. So for example, in height, using 250,000 samples, there were 700 genome-wide significant loci. The H square that I get from my regression on the height similarity of siblings, cousins, and so on and so forth is 0.73. So I would expect to be able to capture 73% of the phenotypic variance if I had perfect understanding of the genetic contributors. But in fact, when you add 700 loci that are genome-wide significant, you end up with only 16% of that variance, okay? And that huge discrepancy between how much I can explain from genome-wide significant loci and how much I could theoretically explain based on other estimates of sibling and cousin and, you know, monozygotic and dizygotic twin similarity, then, you know, I'm far, far off. And the worst part is that these 700 genome-wide loci, if you rank them by how much of the phenotypic variance they explain, our ability to discover them is directly linked to that phenotypic variance. Because our ability to discover them both depends on their frequency and their effect size, okay? So if you keep going down that list of how much I can discover, the 700th variant explains a tiny little fraction of the heritability. So that basically says, well, you know, there's something's got to give. So what are some possibilities? One possibility is that we're in fact truncating that scatter plot distribution of what we can discover with GWAS. Why? Because on the effect size, yes, certainly I need a lot of people, but also on the allele frequency size, I also need a lot of people. Why? Because rare alleles are simply not going to be detectable in a population sample of, you know, 150,000 people. If this variant is only 1 in a million, then I have almost 0 chances of hitting this, okay? So, and certainly hitting this with sufficient number of samples from both cases and controls or across different height regimes, basically levels, to be able to detect a reliable association, okay? One possibility is that there are simply many, many variants that are of tiny, tiny little Manolis frequencies and that have huge, huge effects. Okay, and these variants are simply so rare in the population that I just simply don't discover them. But another possibility is that simply that tail that sort of has to go in these diminishing additive effects from 16% all the way to 73%, that basically means that that tail must be extremely, extremely, extremely low. Okay. Other effects, other possibilities is that simply, we're fooling ourselves just because we're looking at, only additive effects, and in fact, interaction effects are very common, or dominance effects are very common, and so on and so forth. Okay. Another possibility is that we simply haven't finished cataloging all the genetic variation in the human population. So basically, when we're looking at GWAS arrays, we're only capturing common haplotypes. But in fact, there are rare variants within these common haplotypes that are hidden within there. And what we're capturing as the effect of this haplotype is in fact grossly underrepresenting the true effect of the driver SNPs within that haplotype that might be rare SNPs that are tagged within that larger haplotype. Okay? Who's with me so far? Raise your hands. Awesome. Who feels that they've learned something? Great. Awesome. Any questions? Yes. Yeah. There's a lot of studies have basically looked at, that aspect. Basically, if two twins are separated and one lives in a different environment and so on and so forth. So you have to account for that somehow. But you're absolutely right. Maybe we're basically one possible solution to this discrepancy, to this missing heritability problem is maybe we're totally underestimating the effect of the environment. Maybe when we're looking at cousins, they're still in the same city or they're still in the same country. And in fact, the healthcare or the nutritional programs in that, or the pollution, the environmental, components, of that region of the world has a huge effect on that. So that's one possibility, that you can control for that using these types of studies of twins separated at birth and stuff like that. Other questions? Yeah. We believe so. So basically, yes. So the problem there is that we're only, in this particular example, looking at genome-wide significant loci. And in order to reach genome-wide significance, you have to adhere to some very stringent thresholds. We're cropping a lot of that tail. We're going to look at methods throughout the lecture for not having that tail, not having this requirement that we're only looking at GWAS variants. And then when you start looking at all of the variants in the genome, that's when H square G will basically match H square A, basically the additive component. Any other questions? Great. It's too complicated for now, so a lot of people are ignoring this. Any other questions? Great. All right, so basically we talked about the sources of missing heritability. There's still a huge ongoing debate about several possible explanations for the missing heritability problem. Many common variants might be around that have small effects. As I mentioned, that tail could be extremely, extremely long. There could be unobserved rare variants within these haplotypes that have very large effects, or simply our models could be wrong. And every one of these possibilities has very different implications for what we should be doing in human genetics. So let's now start looking at sort of the practicality of all this. We talked about how we can partition variance, how we can estimate the variance, the difference between narrow sense and broad sense heritability. Let's now see how we can use heritability to gain insight into the genetic architecture of complex traits. So there's a lot of evidence that the many common variants possibility is in fact explaining a lot of this. And this evidence comes from polygenic risk scores, linear mixed models, variable selection in our regression, and then forward simulation of risk loci. We're going to go through those. So first of all, you can basically carry out a prediction of your phenotype using many, many genetic loci. So that is known as polygenic risk score. So risk, because this is your predisposition to a particular trait, and then polygenic, because this is actually including many, many genetic components. So this is the aggregate burden of not just the GWAS SNPs, but also subthreshold SNPs, and how these can actually improve prediction performance. So as you include more and more SNPs in the risk score, your association with all of these different diseases gets stronger. And in practice, this requires tuning of the p-value threshold, some kind of LD pruning threshold and so on and so forth. So here's what the data looks like. So basically, if you look at, all GWAS variants that are at 10 to the minus 4 or greater, then your ability to predict rheumatoid arthritis is, relatively small. But as you start increasing the number of variants that you include, if you go to 10 to the minus 3, if you go to 10 to the minus 2, 5 times 10 to the minus 2, 10 to the minus 1, and so on and so forth, you basically see that that ability to actually predict rheumatoid arthritis is, increasing dramatically from that. And celiac disease, same thing. You basically have 10 to the minus 3, 10 to the minus 2, and then you keep increasing. And same with myocardial infarction or coronary heart disease. So you basically see that even as I'm including variants that, I would be ludicrous to consider at 10 to the minus 2, 5 times 10 to the minus 2, and so on and so forth, heritability increases rather than decreases, which basically suggests that there's something going on here in these, very weakly associated variants. But in fact, there's signal, 6 orders of magnitude away from this 10 to the minus, 5 times 10 to the minus 8 threshold of GWAS genome-wide significance. Everybody with me on this one? So the way to include all of these is to basically start developing these linear mixed models. That is basically saying my phenotype is some kind of, predictor from a normal distribution that's zero-centered. This is the, variability in the, say height, the difference from the mean, which is, some kind of heritability from genetics and, 1 minus h squared from, other components. So this is a joint model of all the SNPs, which basically, has been shown to, in fact, explain much more heritability. And the idea is that under some suitable assumptions, the additive phenotypic, the additive component of the phenotypic variance is the sum of all of the betas. And under this infinitesimal model assumption, which basically says that the beta j of each variant is following some kind of normal distribution, which is zero-centered and has standard deviation h squared over p, which basically comes from this equation here. Then you can estimate the additive component of your genetic variants without even estimating the individual beta j's, using only the residual maximum likelihood or residual maximum likelihood, REML. So what residual maximum likelihood does is it avoids using a maximum likelihood fit of the parameters, which would be very dangerous in a way, because it basically, we simply don't have the power to estimate each of these parameters with any confidence as you get to lower and lower and lower p-value associations. Instead of using a maximum likelihood fit, it uses a transformed data so that effectively this nuisance parameter of this maximum likelihood fit is simply having no effect. And in variance component analysis or this random effects model, this transformation focuses on the differences rather than the actual values, and then the sum of the variances. And then this actually works despite actually not knowing what the causal variants are. So number one, you don't have to know what the causal variants are. Number two, you don't even have to know their actual maximum likelihood estimate of the betas. So you can basically do that by effectively using this residual maximum likelihood approach that you can read more about. So what that does is when you apply that to height, it basically goes from 16% of the phenotypic variance that can be explained by GWAS significant loci, all the way to 50% of that phenotypic variance. So that basically says that it is not some rare variants that are sitting on the same haplotype. It is not some rare coding variants that we may have to actually sequence. It's not de novo variant. It is in fact within the same GWAS variants that we have in our arrays. That all of that additional heritability is in fact sitting right there in the genome and right there in the common variants and right there in the common variants that we can actually capture in relatively small population samples. Okay, so that's not quite at 73%, but it's in fact getting a lot closer. And in fact, as this line of work has continued, as the cohort sizes have increased, and as we have better estimates, we are now starting to close the gap between H squared G and H squared. Okay, so that basically suggests that these other explanations of dominance effects or interaction effects and so on and so forth might actually never have to be fully measured, that we can actually get away with effectively just having a very good estimate of these very large numbers of contributing SNPs. Yeah. Oh, sorry. Thank you. Mm-hmm. Correct. Any other questions? So we can basically generalize these regression of relatives, this Haseman-Elston regression that was basically looking at monozygotic twins, siblings, and so on and so forth, to estimate heritability for completely unrelated individuals using linear mixed models. So then the intuition is that the genetic relationship matrix, big G, that is effectively telling you what fraction of genotypes do I share between any two people, captures the identity by state in unrelated individuals. This is the probability of sharing the same allele at the causal variance. And that's called, PCGC regression, which basically says phenotype correlation versus genotype correlation. Okay, that's exactly what you were doing before when you were looking at, siblings, cousins, and so on and so forth. You were basically doing a regression between how much of my genetic variation do I share versus how much of the phenotypic variation do I share. Okay, so this is basically PCGC regression, so phenotype correlation versus genotype correlation. And this is basically saying how much of my genotype correlation do I share in the x-axis, and how much of the phenotype correlation do I share on the y-axis. Okay? So I can start doing this across pairs of individuals and start estimating that, that component. Okay. So what's really cool with this is that I can run it across the entire genome to basically simply say how much of the genome do I share, or I can actually partition it in different subsets of the genome. And if I could somehow magically figure out all of the functional elements of the human genome, I could just simply look at PCGC regression, where the GC, the genotype correlation, is only calculated in the subset of the genome that is, functional, or in the subset of the genome that is in enhancer region, or the subset of the genome that is in DNA scapper-sensitive region, or the subset of the genome that is active in embryonic stem cells, and so on and so forth. Okay? So instead of basically doing this phenotype correlation across, genotype correlation across everything, I can just do it in different subsets. So who's with me on PCGC? On basically the concept that I can estimate this H square as the slope of this phenotype correlation versus genotype correlation scatter plot. Okay? And now, who's with me on the partitioning this GC component into the different parts? So raise your hand if you're with me so far. Awesome. Any questions? Who's completely lost? Okay, good. No one's completely lost. All right, good. So let's try that. So we're now going to start partitioning heritability. We're going to take this GC component and basically look at different fractions of the genome. So I see some people laughing. So please raise your hands if you're with me on the PCGC part, on the sort of slope that relates phenotype similarity versus, sorry, genotype similarity on the x-axis versus phenotype similarity on the y-axis. That slope effectively tells you about heritability. The closer I am to, a straight line, the more this is, purely driven by genetics. And then the further I am, the, environment basically has a bigger component. Basically, that relationship effectively defines another measure of heritability. So in this equation, it's basically, this heritability is relating the phenotypic similarity to the genotypic similarity. Raise your hands if you're with me on this. Awesome. All right, so basically what we're gonna do now is actually start chopping up our genome in different ways and basically seeing how much heritability do I capture, okay? So what you can basically do is effectively do your PCGC regression on, chromosome 2 only or chromosome 1 only, and so on and so forth, okay? So as I start chopping up the genome by chromosome, what I'm basically finding is that the fraction captured, this PCGC, estimate of heritability captured in different chromosomes is in fact almost directly related to the length of that chromosome. So human chromosomes have been ordered by, have been numbered based on their length. So chromosome 1 is the biggest, chromosome 22 is the smallest, okay? So you can basically see that, in fact, this is almost a straight line. So, that basically, so you can extend the model so that chromosomes can explain different proportions of variance. You can add more variance parameters to each partition of the SNPs. Each partition induces a different genetic relationship matrix. So I'm only looking at the similarity in genetics for that subset of the genome. And longer chromosomes, turns out, explain more heritability. So what that suggests is that, in fact, causal variants are spread out uniformly across the genome. That, the more area I look, the more heritability I will explain. Which is basically saying that, goodness, there's a lot of them. There's a lot of genetic variants. And you can basically ask, how is this, working for different traits? If you look at dyslipidemia, hypertension, schizophrenia, you can basically, look at the fraction of the genome that is contributing to each of these traits, the H square for every 1 megabase window. What you can basically see is that, yes, for some traits, like dyslipidemia, there is a lot of heritability that sits in a small number of loci. But as you start getting to hypertension and schizophrenia, you basically see that this heritability is, in fact, pretty uniform across the genome. That, so you can feed a model with one component for every megabase window, and that was done by Rouleau. And you can bound the cumulative heritability explained to estimate the number of regions. And what you can basically see is that, in fact, most of the genome explains non-zero heritability. So basically, these variants are everywhere. You can also basically start selecting variables in a more, yeah, go ahead. Yeah, so we didn't look at those. Yeah, in a way, because of the chromosome sex inheritance patterns, they're much harder to model. So a lot of the time we're going to be focusing on autism. So what you can also do is start selecting subsets of the genome in a more interesting way. So for example, you can directly fit the underlying linear model so that only a very small subset of your genome has non-zero effects, for example, and then you can estimate that effect. So if I can start partitioning my genome into different functional categories, for example, I can then ask just how much of that can I model using this. So I could actually inform these pathways directly from the enrichment. So basically, some pathways contain more causal variance than the rest of the genome. So what I can do is actually incorporate that into the prior. So basically, I can calculate an enrichment of different pathways. And then once I have my enrichment, I can use that to increase the prior of particular pathways. And that can identify, for example, immune signaling pathways for specific immune disorders, which are not captured using existing methods. And the reason is that they just, they have a lot of genetic variants, each of which has very small effects, so you wouldn't be able to capture them. But when you partition heritability accordingly, then you find, effectively tens of thousands of SNPs that could be affecting these pathways. So you can turn this on its head. And this is what Agarwal et al. Did in 2012. So she basically said, let me simulate in a forward fashion, rather than reading what evolution gave me in a sort of reverse engineering fashion. What I'm going to do is simply put some parameters in the model, simulate it, and see what that gives me. So she basically simulated realistic loci using known, population parameters and evolutionary parameters. And then she simulated disease phenotypes by varying the number of causal loci, the heritability, the prevalence, the strength of the selection. And then she performed twin studies, genome association studies, and then she compared the predicted results to the observed results on real data. So she basically said, here's, for example, the sibling risk. Here's the, affected sibling pair genome-wide linkage. Here's, the GWAS that I would obtain. Here's my replication. And here's what I actually see in the data. And then, are these results consistent with my simulation? So what you could basically do is distinguish effectively which of these parameter settings were consistent or inconsistent with the observed data. And she could basically say, well, suppose that this is the sibling risk, or the sibling risk is higher or lower, and so on and so forth. And then, what is the, genome-wide linkage, or, what does the GWAS look like, and so on and so forth. And she could basically say, well, here, for this particular setting of parameters, there's several inconsistencies. For this one, well, these things match, but these other guys are inconsistent. And then for this architecture and that architecture, then they are consistent with the data. So what was nice with that is that you could actually say, sorry, some of these are simply inconsistent with our data, and then narrow down the space for of possible GWAS architectures. Okay, who's with me so far? Awesome, great. So there's a lot of evidence for other explanations as well. For example, you can incorporate identity by descent in unrelated individuals. You can partition SNPs by minor allele frequency or by LD, and then some of these assumptions do not hold in real data. So there's another way to estimate heritability, which is shared haplotypes. So basically, shared haplotypes can explain more heritability than tag SNPs. And the reason for that is that the tag SNPs only explain those SNPs that are high enough frequency and that were, in fact, defining these phenotypes. They are the early SNPs that happen, very early in human population history, and that are now defining these haplotypes. But in each of these haplotypes, which were defined early on, additional SNPs have basically creeped in additional populations. And every population has basically had a large number of rare SNPs added on top of that background. And those rare SNPs are sometimes less subject to selection because, they are rare enough that they can escape selection, or sometimes they're in a bottleneck population and everybody has it, so you don't have as, much of a deleterious effect. So what you can do, however, is basically say, if I estimate not just the impact of a single SNP, but the impact of a haplotype based on the sharing of that haplotype by understanding the population history of these two regions, then I can actually account for the rare SNPs within that haplotype. Who's with me on that concept? Great. So, the shared haplotypes can basically explain more heritability than the tag SNPs, but, the concept for that is that you can actually calculate identity by descent by basically saying, well, I can infer which both common and rare variants I have inherited from that common ancestor using, in this particular region, using both the relatedness of these individuals and the haplotypes that they've inherited. So basically, the idea here is that if two individuals share a chromosomal segment, the unobserved variants in that segment should also be shared. And then the idea is that you can identify identity by descent segments by quickly scanning SNPs and finding stretches of identical alleles. You can infer shared segments and these inferred share segments capture rarer variants, much more effectively. And, you can increase this, H square that you can explain from these segments, okay? But there's still a discrepancy between H squared G and G. The other thing you can do is basically ask, at what range of my minor allele frequencies do I have, more heritability? So what you can do is partition your heritability for different minor allele frequency ranges. So these are very common variants, slightly less common variants, rare variants, super rare variants, and then private variants. And then we can basically ask is, how is my heritability changing as I go further and further, okay? And then the low frequency LD variants are poorly tagged by observed imputed variance. So the estimate, their variance is estimated separately. And then partitioning appears to explain all of the heritability of height using only common and low frequency variants. So that basically suggests that we don't have to go to the super rare variants to explain more heritability, that basically a lot of them are in fact sitting right there in front of our noses. It's just that they have very, very small effects. So you can start examining the model assumptions. The phenotypes might actually not be Gaussian. So, that's a problem. So the GWAS samples are not independent and they're not identically distributed. Basically, there is in fact cryptic relatedness in a lot of the GWAS samples. The SNPs themselves are not independent. Not all SNPs have an effect. Doing these infinitesimal models, we're basically assuming that you can actually calculate these effects. Not all causal SNPs have equal effects, so the effects of some SNPs might actually be stronger. There are in fact some gene-environment interactions. There are places where the environment and the genetics are coupled. If you come from particular regions, you're likely going to share both the environment and your genetics. And there are in fact gene-gene interactions. So basically all of that might actually contribute to additional heritability. There are also a lot of limitations of heritability. So explaining all of the heritability of a complex trait is not enough. Basically, as the sample size goes to infinity, the big question is, will the entire genome be associated for all traits? And we're going to talk a little more about that soon. And then, maybe the goal should be to find biological pathways that are recurrently disrupted by these variations. And that's where a lot of the earlier lectures come in. So basically, when we set out to study the non-coding genome, we defined all these classes of elements. For example, if you look at the roadmap Epigenomics project, we basically have enhancer regions that are active in different subsets of the cell types. So you can actually start asking now about the partitioning of that heritability in different functional classes. And the first glimpse of that came from looking at ENCODE annotations and then going down the association score and basically asking, where do I see functional enrichments as I go down that score? And what Morano showed in 2012 is that if you look at, for example, the QRS interval, this heart repolarization direction between the Q, the R, and the S, landmarks of this heartbeat, then what you can see is that as I go down the p-value association from 10 to minus 8, 10 to minus, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, etc., I basically continue seeing that genetic regions are preferentially located in epigenomic annotations that are active in the heart, even as I go down to very, very weak associations. So that basically says not only are these rare variants, not only are these very weak effect variants continuing to contribute to heritability, moreover, they continue to localize in the same functional categories as the genome-wide significant variant. If you look at Crohn's disease, you basically find that T cells are, very strongly enriched, and they continue to be the top categories much, much further down the rank list. And same for multiple sclerosis and so on and so forth. So that basically suggests that, these guys are continuing to function in very similar ways, that this is not something where the top variants are doing some things and then the bottom variants are doing other things. In fact, the same processes that we can discover in the top variants continue to be relevant as you go further down the rank. Who's with me so far? Yeah? Any questions? So then what you can basically do is build a joint model of SNPs and annotations. So you can basically use penalized stepwise regression to pick relevant annotations, and then use approximate Bayes factors to compute the posterior probability of association. And then you have a backward step and a forward step. In the forward step, you add annotations to the model until they don't explain enough variance. In the backward step, you remove annotations from the fitted model until the variance explained drops too much. You can basically select annotations this way. You can see how enriched are they. Can I add them to my model, and do they, hurt my association? And when you do that, you can basically, prioritize GWAS loci that are consistent with your annotations and also consistent with your genetics. So that basically allows you to sort of now prioritize new loci. You can also, in the same way that you could sort of split your genome by cell type, you can now split your genome by different classes of annotations. You can basically say, well, let's take all coding annotations, all untranslated regions, promoters, intronic, intergenic, and all DNA's hypersensitive sites. And then ask for each of them, what fraction of the SNP heritability can I explain using these annotations? And how enriched is it over what I would have expected by chance? And if you look at intergenic regions, I would have expected that they capture about 55% of the heritability. This is the dotted line. And that basically is simply because they capture about 55% of the genome. But in fact, they're very severely depleted for explaining heritability. If you look at intronic regions, same thing. You would expect them to capture a big chunk, but they're in fact depleted. If you look at coding, UTRs, and promoters, they're dramatically enriched compared to what you'd expect. You would expect coding regions to only capture 1% of the heritability because they only capture 1% of the genome. But in fact, they capture about 7% of the heritability. That basically means that coding regions are very, very enriched for heritability. They're much more likely to capture functional elements. Same for UTR, same for promoter regions. But the most dramatic result is that DNA's hypersensitive sites, which are expected to capture only about 18% of the heritability, in fact capture 80% of the heritability. So that basically says that the vast majority of heritability for these complex traits is in fact sitting right in front of our noses in DNA's hypersensitive loci. That basically using this epigenomic enrichment to understand where are these regulatory loci results in SNPs which capture much more of the relatedness that matters between individuals, where the phenotypic relatedness and the genotypic relatedness are in fact much more aligned when you focus on common relatedness of SNPs in VHS. Yeah. Taylor? What are they? So these are the accessible regions. So if you do, if you recall the epigenetics lecture at the beginning of the lecture, I basically showed the 3 different types of epigenomic information. So one was DNA methylation, one was histone modification, and one was DNA accessibility. So DNA's hypersensitive sites are basically regions of the genome that if you throw DNAs, it will be able to cut them. That basically means that these regions were not protected by nucleosomes. These regions were accessible. It's another way to say accessible regions. Yes, I see what you mean. So first of all, across species, they overlap genomic regions that are preferentially conserved between species. And DNA hypersensitive sites are, to a great extent conserved between people. So basically, one criticism could be that, hey, we only looked at one person when we looked these DNA hypersensitive sites. Maybe they vary from person to person. So maybe we shouldn't be focusing on only on one person. But in fact, a lot of them are conserved between different people. And this particular example is looking at, adding up many, many different types of cell type annotations. So that suggests that, in fact, we are capturing a lot of the DHS, and there's the different cell types. or sometimes sampled from different people. So you also capture genetic variation there. Mm-hmm. So if you look at the confidence intervals, they're basically more or less centered around 0. So, you know, you can't quite argue that it's more or less, but it's, you know, it is funny that it falls below. Yeah. Okay. So basically, if you combine these accessible DNA's regions, more than 100 cell types, 70% of the genome is accessible, but only 16% is accessible in multiple cell types. In this particular case, you basically see the 16%, which is found in at least 2 experiments, you know, captures a lot more than that. So that basically implies that non-coding SNPs explain a lot more variance per SNP in coding SNPs. So then you can go a step further and basically say that if I have a genetic variant that sits, you know, in this longer haplotype block, and if I have a genetic variant that sits in this smaller haplotype block, the one in a longer haplotype block will basically have many, many different other genetic variants that it also captures. And if, in fact, I have many, many very densely, you know, spread out SNPs that are all contributing very weakly to the disease, the larger my haplotype block, the more of the true contributors to the disease I will capture. And therefore, if I basically have SNPs that are highly correlated with each other, each of which is in fact capturing the inheritance patterns of additional nearby SNPs, then I will expect to capture more heritability in these regions. So the intuition here is that causal variants are drawn uniformly at random from the genome, and these are more likely to come from larger LD blocks. Okay, is this intuition clear with everyone? That basically now that I have all these super densely sampled causal variants, larger haplotype blocks are more likely to contain more causal variants. Everybody with me on this? Yes, raise your hands if you're with me. Okay, awesome. So that basically also means that if these larger blocks have more causal variants, that means that any one variant in these blocks, because it captures the inheritance pattern of multiple nearby SNPs, is more likely to, in fact, have more effect, basically bigger association. So that basically means that, you know, these genetic variants are likely to be, you know, upweighted, if you wish, than those genetic variants. So basically a linear regression of the summary statistics against the LD scores would effectively give you H square, this heritability, without access to genotypes. And then the idea for this is that this H square, this heritability, is directly associated to the density and the larger number of these genetic variants, which is a fraction directly of the LD patterns in that region. So then you can use this approach. You can use this heritability using this LD score regression. So basically this LD score regression can basically be generalized to partition it into the multiple components. So then you can take all annotations from the roadmap of genomics project and then split them into non-overlapping sets. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different groups, which basically group multiple cell types and multiple tissues. So adrenal or pancreas, central nervous system, cardiovascular, connective or bone, gastrointestinal, immune or hematopoietic, kidney, liver, skeletal muscle, and other. So now you can actually start partitioning these annotations, and every single SNP only belongs to one of these annotations. And we can basically ask is, if I take the heritability of schizophrenia or bipolar disorder or height and I partition these SNPs into the different annotations, then which of these partitions is enriched over what you would expect by chance given its coverage of the genome. And you can calculate a p-value for that enrichment. And what you can see is that if you look at different diseases, you basically find that very different components are in fact very strongly enriched. If you look at schizophrenia, you find central nervous system. If you look at height, you find connective tissue or bone. If you look at rheumatoid arthritis and Crohn's disease, you find immune and hematopoietic components. Look at fasting glucose, you find adrenal components. If you look at BMI, you find central nervous system, huge component in eating, for example. Years of education, again, central nervous system, whether you smoke or not, central nervous system. If you look at the age of menarche, basically when you start having your period, you basically have both central nervous system and adrenal components. If you look at cholesterol, you basically find liver and so on and so forth. Okay, so that basically means that I can use partitioning of heritability to understand where is most of the heritability across not just GWAS significant, not just subthreshold, but across all variants in the genome using this LD score regression approach. Who's with me so far? Raise your hand if you are. Awesome. And that basically takes us to sort of the most recent developments in this space, which is basically saying, you know, yes, indeed, heritability seems to be clustering in these top annotations that make a lot of sense. But then look at all these other annotations. They're all continuing to be, you know, enriched. So, you know, what is the matter? Asked basically Alan Boyle, Jonathan Pritchard, and Lee. And what they basically said is, if I estimate the genome-wide enrichment in this QQ plot for, you know, simply having SNPs, what you can see is that as I increase the number of individuals, this QQ plot is no longer tracking this line all the way to 10 to the minus 4, 10 to the minus 5 before it reaches genome-wide significance and boosts off the line. It in fact tracks it very nicely here at the bottom, suggesting that my model is in fact well calibrated. But then it deviates very early on at 10 to the minus 1 and 10 to the minus 2. So that basically suggests that there is a very large number of these subthreshold variants that appear to be both enriched genome-wide, but also specifically localizing within, say, EQTLs or, you know, enhancer associated histone modification markers. I can also simply ask, you know, just how much of that heritability do I capture at different LD scores, or how much heritability do I capture at different levels down the p-value? And what you can see is, for example, this replication effect between different studies doesn't just stop at the genome-wide significant loci, but at, you know, 10 to minus 4, 10 to minus 2, and so on and so forth. It continues to, basically at p-values of quarter, p-values of a half, p-values of 0.75, it continues to replicate above what you would expect by chance. Basically, what they reason is that my replication frequency would be down to 0, you know, after I start having signal in my GWAS. But in fact, I continue having signal all the way there. So that basically suggests that in 2 different cohorts, the genetic effects estimated are consistent, which suggests that, in fact, I have the power to estimate these genetic effects very, very far down the list of genome-wide significant loci. And in fact, what they pointed out in addition is that these heritability is not only concentrated in very disease-relevant loci, but also in very broadly expressed loci. So basically, even though the fold enrichment for heritability is greater in frontal cortex for schizophrenia, the highest amount of heritability is greater for these broadly expressed regions, suggesting, in fact, that you know, that it's not only these core disease processes, but that these core disease processes are certainly the most enriched for heritability, but they don't explain most heritability. And in fact, there are other processes nearby that explain more heritability. And if you look at gene ontology categories, what you basically see is, here's what I would expect on the x-axis for each Go category based on the proportion of SNPs that they have. And then here's what I find in terms of the proportion of heritability that they capture. And all of the Go categories, or nearly all of the Go categories, are above that diagonal, suggesting that every single Go category is more explanatory than what I would expect by chance, which is, very weird. So the model that they're proposing is that, in fact, there's a core set of genes that have the strongest effects and the most enrichment in heritability, but that these guys are only capturing a tiny little fraction of the heritability. And as you start going out from these core genes to more and more and more of the genes in the genome, you capture more and more heritability, but that heritability is no longer concentrated in these core processes. And in fact, these auxiliary processes that are very tangentially related to disease are dominating in heritability, and that in fact, this is true for every trait, and that these auxiliary processes are effectively capturing the rest of the genome. And therefore that pretty much everything matters for everything. So every SNP matters for every trait, which, is a little hard to capture because, and to conceptualize, because in a way we justified GWAS by basically saying we're going to find the genetic drivers of disease. And now if we go back, sorry, and say we found them and they're everywhere and they're all of them for every single trait, it's a little problematic. So this is sort of where the field is in terms of basically realizing that yes, there is a core, and yes, most of the enrichment is in the core, but in fact, most of the heritability is outside that core. Who's with me so far? Raise your hand. Awesome. Who's totally depressed? I'm kidding. Even you shouldn't be depressed, because in fact, that core is extremely biologically relevant, and that's basically what's going to guide a lot of our disease studies, and in terms of mechanism, in terms of intervention, in terms of therapeutics. Yeah. Enrichment. Yeah, so enrichment in heritability basically says that even though I capture only 1% of the genome, I explain 3% of the heritability, right? That's a threefold enrichment. That basically says, wow, I explain a lot more heritability than what I would have expected given how much of the genome I covered. But we're still at 1% and 3%. Then there's these other guys that cover, 70% of the genome, and they explain 40% of the heritability. That means they're depleted for heritability. Great, they're the things I can ignore. But what you can't ignore is the fact that there's another 40% to be explained, and they are the guys explaining it. Does that make sense? So it's basically a question of both the numerator and the denominator. The numerator, the ratio of how much I explain versus how much I would have expected to explain is very high for the core gene, and they fall in the functional process you would expect. But then the numerator itself matters. And that numerator, even though there's a threefold enrichment, is only, 3%. Whereas the numerator for the other one, even though it's depleted, is still 40%. That's a great question. So basically what you're saying is, how can they all be enriched? Yeah, does that mean that there's an ascertainment bias in creating these Go categories? And in fact, there's a bunch of genes that are simply not in any Go category, and they are the boring genes that don't do much. That's one explanation. Another explanation, in fact, there's many processes that are below, and these processes can sometimes be much larger. And that, they account for these variations. But I think your intuition of maybe there's an ascertainment bias of the things that have the bigger effect, and that's how we found their function in the first place. Maybe accurate. Okay. Everybody with me so far? So basically now, yeah. These guys here. This is basically some pictorial representation of a regulatory network and the interactions between different genes. So basically, you can think of every dot as a protein, and, the different colors as the proteins that are the most associated with the function of that process. And then, some kind of effect as every edge. it's a pictorial representation, but it sort of suggests that all of these guys are contributing to the disease, but through these core genes. That basically there's a network of contribution, and it's sort of, they're not touching these core processes directly, but, there's a gene involved in, I don't know, methyl transfer, DNA methyl transferase. And then that gene is here. It doesn't have a function directly, but one of the genes that it impacts is some regulator. And that regulator is not involved directly either, but one of the genes that it impacts is this core gene in sort of the transfer of calcium in your synaptic connections or something. And, all of these processes are then going to be involved in many, many other things, but they also contribute weakly to the core set of genes. All right, who's with me so far? Great. Do you feel like you've learned stuff today? Awesome, good. So let's now basically see how we can use these to sort of learn more about the biology of these processes. So this is a ranking of all of the SNPs, and they're ranked by p-value, and this is their cumulative deviation in the enrichment for different biological processes. So what you can basically do is say rank all of your SNPs based on their association with type 1 diabetes, and then ask how biased are they for epigenomic annotations in immune processes. And we can basically see here that there's this huge enrichment, and then, you sort of fall back down. But this huge enrichment spans a million SNPs. So that suggests that, again, this goes very, very far down the rank list. And this can be seen in many traits. So basically, if you look at CD, Crohn's disease, rheumatoid arthritis, type 1 diabetes, Alzheimer's, and so on and so forth, you basically continue seeing these enrichments across, many, many, many loci. So here, what Abhishek actually did is resolve the SNPs that were completely independent from each other. And then still there are thousands of independent loci that contribute to each of these traits. And then he basically said, well, enhancers are sometimes active in, basically all the enhancers that I call as active in these immune tissues, for example, can be also found active in other tissues. So what he did is actually cluster enhancers into modules of enhancers that are coordinately active. And what he found is that these modules are partitioning effectively the genome into distinct functional classes, including this common module. And what he found is that these enhancer modules very strongly enriched for a number of these traits. And it wasn't just the immune modules, it was also these constitutive models that were leading to a lot of enrichment. Again, sort of consistent with this concept of these broadly active genes involved in many, many diseases, even though they're not just active in that particular cell type. And then he was able to sort of go from these genes to the corresponding, from these enhancers to the corresponding genes, linking them together based on, multiple lines of evidence, and then getting at sort of hundreds of genes associated with each of these traits, suggesting that in fact, there are many, many novel genes in each of the enriched pathways. And then he used these common pathways and this large number of genes, basically look for upstream regulators for each of the traits. So basically for every trait, he said, well, here's the core regulators that are sitting upstream of all of these genes and all of these pathways. And then that sort of gave rise to, this list of master regulators that are now controlling, exerting an impact on many, many of these genes. In a way, this is sort of directly related to this omnigenic model with a core set of genes, and now saying that all of these genes that are contributing to heritability are in fact sometimes targeted by master regulators that are sort of sitting elsewhere in the genome. And then he could basically build these, regulator to gene networks across many different diseases. Every edge is now a disease where, and every regulator is sort of these hubs, and all of the target genes are nearby. So you can now start explaining how all of these different regulators are exerting this global influence. And then what he found is that many of the predicted master regulators were in fact in constitutive enhancers rather than cell type specific regulators. And then although these enhancers were marked as constitutive, the upstream regulators were very cell type specific. So that could actually start explaining how we can get cell type specificity of the action, not through the activity pattern of the enhancer, which is the cis acting component, but through the activity pattern of the regulator, which is a trans acting component. And then lastly, we can use all that to now start asking, how can I predict phenotypes? And we talked about these, imputation-based models for association. But now you can think of these not in the context of a small number of genes, but in the context of the entire genome. So now you can start building these linear mixed models that allow you to now start predicting the phenotypic output for all of these intermediate phenotypes using the full matrix of SNPs and using all of these contributors, all of these betas, basically the effect sizes, which you're now sampling from a distribution. So now you can estimate the overall phenotypic output for every one of these variables using the contribution of a very large number of genetic loci, each weighed by its effect size, and some random noise associated with each of that. And then you can optimize this model to infer what are the drivers based on the genetic relationship matrix, and then estimate that using these maximum likelihood models. So you can basically do variable selection to select the loci that are the most likely to be contributing to the disease. You can use these regularization approaches, L1 regularization and L2 regularization, to basically limit the number of predicted variables, and then end up with the set of SNPs that are predicting your phenotype in this extremely polygenic network. Basically, you can look at one SNP per locus, you can look at all of the SNPs, and you can sort of model the selected set of SNPs, and then boost your power by effectively selecting the relevant SNP in each of these loci. And you can do that by basically having these spike slab models that basically have a spike at 0 and then a slab for the surrounding one. And then that spike is specifically the SNPs that you've selected, and then the slab is all of the other SNPs that have relatively smaller effects. So what we talked about today was this fundamental concept of heritability, basically how we can partition phenotypic variance into genetic and environmental components, how we can partition genetic variance into the independent, the dominance, and the additive components, the difference between narrow sense and broad sense heritability, and how that can allow us to actually study the genetic architecture of complex traits by including polygenic risk scores, linear mixed models, partitioning heritability, and then this omnigenic model of complex traits, and how we can go from understanding genetic architecture to the systems biology connecting these enriched loci to the genes, the pathways, and ultimately the regulators. And then very briefly, how we can talk about phenotype prediction by basically imputing these intermediate phenotypes across very large numbers of predictors. That brings us to the end of this module. And then next week, we're going to talk about evolution. Thank you all.